There are no direwolves south of the wall.
That is a woolly mouse, a new kind of mouse created using gene editing.
This experiment started off with collecting DNA from our pet dog here, and also taking DNA from our pet rabbit.
It's an animal that hasn't existed for some 13,000 years.
Where we took them to our laboratory and quickly spliced them together so we could fertilize eggs. After letting the eggs sit in the incubators for many months, they began to hatch.
But wait! There's more! Hang on to your seat, baby! Cause this one's a screamer!
And the results were breathtaking. As you can see here, we have these small white dogs, but they look like bunnies. They are half bunnies. They're very jumpy and fun and happy. Should we call them bogs or dunnies?
To give it woolly mammoth-like traits. The reason that they're doing this is they want to try to bring back the woolly mammoth, but they're not literally cloning or resurrecting it. Instead, they're trying to edit existing animals' genes to make them look more like a woolly mammoth. Specifically, they plan to do this with Asian elephants, their closest living relative. And along the way, they created the woolly mouse by analyzing preserved DNA from woolly mammoths to pinpoint genes that made them suited for colder weather, and then looking for similar genes in mice. Using gene editing tools like CRISPR, scientists edited 7 mice genes all at once. And the mice turned out with these thicker, wavier coats and a slower metabolism. They didn't insert any new genes into the mice. They just combined a bunch of genetic changes that they'd seen in individual mice into a single mouse. We have an unbelievable story coming out about gene editing, so if you're interested, follow along.
Now there are 5.
But the long extinct species now making global headlines for a new reason. A Dallas-based biotech company saying they've resurrected the giant Ice Age wolf, essentially bringing it back to life through a process called de-extinction.
What is this? What the hell is happening?
Colossal is the world's first de-extinction company, and our goal is to make extinction a thing of the past.
Colossal says they've genetically engineered 3 wolves that they say resemble the dire wolves. The company sharing these videos showing the animals howling as cubs, and here sleeping and running in their habitat at an undisclosed secure location in the U.S.
People think it's a scary word, de-extinction. They immediately put us with Jurassic Park, and it's not. We're trying to develop these new techniques in order to save animals.
But the process does sound like something out of the 1993 blockbuster. The company says scientists relied on ancient preserved DNA from a tooth and a 72,000-year-old skull to decipher the direwolf genome. They then used gene editing technologies to rewrite the genetic code of the common gray wolf.
So one at a time, we take our gene editing tools and we go into that cell and we cut out the bit of DNA that looks like a gray wolf and paste in its place the DNA that looks like a dire wolf.
Once a new genome was created, domestic dogs were used as surrogates, miraculously giving birth to Romulus, Remus, and their sister Khaleesi. Yes, named after Game of Thrones. But this scientific feat, no fantasy, and some online already questioning whether it's a step too far. Still, the technology is here, and scientists say it could shape the future of our ecosystem.
We produce something that is magnificent and science fiction at its finest.
Just amazing. Now, the company says one reason they chose to bring back the dire wolf is because of the importance of predators and helping revive ecosystems. Like in Yellowstone, they say reintroducing wolves help bring back beavers and songbirds. That said, the company has no plans to release any of these dire wolves, which we haven't seen ourselves, by the way, back into the wild. They do, however, hope to breed more of them, and they are also working on other animals, including the woolly mammoth and the dodo, guys.
Oh, they're working on the woolly mammoth. Okay, Liz, thank you.
Thank you. You excited about that?
No, I'm a little. For whatever reason, I'm fine with the wolves, the woolly mammoth. That scares me.
Well, I know.
That
the dodo
scares me.
seems more of a friendly situation.
I mean, we sort of killed the dodo, so that's the least we could do is bring that one back.
For decades, scientists have been experimenting with the idea of changing the human genome to cure genetic disease. Some researchers now say they're closer than ever before to developing a treatment to correct defective human genes thanks to new gene editing technology that could provide scientists with unprecedented power to rewrite the code of life. With the new technology called CRISPR-Cas9, scientists hope to create future medicines that can fix errant genes that cause diseases such as cystic fibrosis and certain cancers. The process involves 2 basic components, the Cas9 protein and a programmable so-called guide RNA. The guide RNA is a single molecule that directs Cas9 to the specific sequence in the genome. Once there, Cas9 works like a molecular scalpel, cutting out the flawed part of the sequence that causes the mutation. A gene-editing drug would include the Cas9 protein and a guide RNA specifically targeted for the mutation. It could also carry healthy DNA to fill in the gap. Scientists say CRISPR-Cas9 is a revolutionary tool because it allows them to target specific genes more precisely than previous gene correction technologies. There are other advantages as well.
The nice thing about Cas9 is it's very easy to use and very inexpensive to use. It doesn't take you years and years and millions of dollars to try your idea. It takes you weeks and a couple of $100s. And so that means that you can be very creative and very broad in the things that you test.
Scientists believe the new technology could find many applications in biomedical research and eventually allow them to create treatments for diseases caused by mutated genes. It may be years until the first human clinical trials are conducted, and maybe a decade or longer until gene-editing therapeutics hit the market. But many laboratories and pharmaceutical companies across the country and the globe have adopted CRISPR-Cas9 since it was first introduced in 2012. Some pharma companies are already exploring how to create gene-editing drugs for certain genetic diseases. Some labs are using the genome editing technique to genetically engineer laboratory animals with genetic mutations and then study the recreated mutations inside the animals. At the Innovative Genomics Initiative in Berkeley, California, scientists recreate diseases in human cells to learn more about how mutations cause illnesses and how they can be corrected. Although many scientists have high hopes for the technology, there are still challenges, they say. For example, CRISPR-Cas9 is generally precise, but it occasionally cuts the genome at unintended sites.
I mean, we're talking like 1 in a million, 1 in 100 million. So Cas9 might come in and it might make a cut, and it might also make a cut somewhere else. And we're still trying to understand how frequently off-targets occur, how much specificity is a problem. As you can imagine, from a therapeutic point of view, we want to know the answer to this.
Scientists say it's also difficult to deliver genetic changes to all the cells in the body that need it. To treat some diseases, scientists will be able to extract blood cells or bone marrow, alter them, and then put them back in. For diseases in more complex regions, like neurological diseases, more research is needed to find out how to get the Cas9 protein to the right spot. There is still a lot more work to be done, but many scientists believe that the Cas9 technology could one day help cure many genetic diseases.
Alright, here's a go-to sci-fi storyline that just keeps on giving. Scientists mess around with DNA, and then crazy things happen.